Listo. Buenas tardes a todos. Ya veo que están aquí conectándose muchas personas, lo cual agradecemos. Estamos acabando el primer día de FEVIE. Y bueno, antes que nada y antes de dar la bienvenida a todos la gente de Truking y nuestros invitados, quiero aprovechar el momento para felicitar, pues no felicitar, sino aplaudir el esfuerzo y los logros que han tenido las mujeres en esta época. De una lucha por la justicia y la equidad, buscando que esto sea más por la capacidad de las personas y no por el género. Y bueno, creo que las mujeres nos han enseñado bastante de cómo se debe luchar. Lo cual, pues mi más sincero respeto a todas las mujeres que nos acompañan y a todas las que están presentes con nosotros, de verdad. Y bueno, ahora sí, doy la bienvenida al evento final del día de hoy. Con mucho gusto, Truking, lo presento, pero antes les quiero dar un poquito el contexto de muchos nombres, y pues, los que quieran y puedan prender su cámara nos daré a gusto verlos. Veo muchos nombres que no conozco, pero bueno, nos presentamos como Rash en esta FEVIE. Y bueno, la intención de esta FEVIE, cuando nosotros la desarrollamos hace 2 años que nos encerraron, estamos cumpliendo prácticamente 2 años que nos encerraron nos fueron encerrando por estados. Y bueno, surgió este concepto y este modelo para tratar de seguir cerca de nuestros clientes. Hablamos con nuestras representadas de qué forma el no viajar podíamos estar con nuestros clientes, y desarrollamos todo este modelo que en realidad es pues platicar algún tema, platicar alguna tecnología que pudiéramos compartir con ustedes. Ya es el tercer evento que realizamos, lo realizamos en el 2020, en el 2021 y ahora en el 2022. Me da muchísimo gusto que lo sigamos pudiendo tener de esta forma y tener el apoyo de todas nuestras representadas, en este caso Truking. Y bueno, pues le doy la bienvenida a Sabrina, que nos hará una pequeña introducción de Truking y del tema para dar entrada a nuestro exponente con un tema muy actual, que es el desarrollo de las vacunas. Entonces, Sabrina, bienvenida y bienvenido todo el welcome to all the team of Truking.
Hola, buenas tardes a todos. Muchas gracias por asistir a esta presentación. Como puede imaginar, con la difusión de virus corona se ha desatado una competencia contra tiempo para desarrollar las vacunas de COVID. Entonces, hoy vamos a hacer una presentación de la solución completa para la fabricación y filtración para vacunas. Ahora presento a mi compañero que es experto, Mr. Pramod, para empezar la presentación. Pramod, you can start your presentation now.
Yeah, okay. Okay. Good evening, all of you. So first of all, I hope you can see my screen as well as myself on webcam. Okay. So I'll start with my presentation. First of all, my name is Pramod Kharatmol. I'm from India and I'm taking care of global business leader for bioengineering division for Asia and Europe. Okay, so thank you for giving me a good opportunity to talk with you all wonderful guys. And on this occasion of International Women's Day, I really appreciate the support all the women are showing and making the world and the society beautiful. Thank you very much. So let's start with the presentation. Today, I will be more talking about, as you know, that COVID-19 hit in 2018 and the world become gone for a turmoil. And because of that, let's see what sort of like a vaccine. So I will be giving you in this presentation, we will walk through different process unit operations required in COVID vaccine manufacturing or in general in any of the vaccine manufacturing plant. So let's start the presentation. So I will be talking about introduction to corona vaccine and basically what sort of different types of them. Okay, and there are various processes required for vaccine manufacturing, like different unit operations involved, like upstream and downstream, where the bioreactors, depth filtration, and for the purification part, there will be chromatography systems and chromatography resins as well. In the second part of my presentation, I will be giving you a brief introduction for our very flagship products for pharmaceutical product formulation, that which involves the pre-filled syringes, inspection instruments, and packaging instruments as well. So in this slide, there you can see a global map, okay, where, for example, like in India, there are different manufacturers of vaccine, and in China, there are multiple COVID vaccine manufacturers. In particularly, if you talk about the Russia, there are like Amelia Research Institute, BioCAD, Nanlake, RusBio, and in UK and in America, there are so many different biosimilar manufacturers who are now involved into the COVID-19 vaccine manufacturing. So this is a very limited list which I have shared on this slide, but definitely the number of COVID vaccine manufacturing or in overall vaccine manufacturing took a very big leap, and throughout the world it is in huge numbers, and all the people are tirelessly working to control the pandemic as well as and helping all the society across the borders of their individual nations. Traditional vaccine development. Let's see an example. So traditional vaccine development involves planning, recruiting patients for and executing each of the 3 clinical phases and generating the data necessary to design each of them. So a vaccine must be shown in the first case, a vaccine must be shown to be safe, that covers in the phase 1, which typically involves 6 to 12 months of data collection after each patient is given a dose. In the phase 2, trials provide an understanding of the dosages required, and phase 3 is full of efficacy study. As companies gain greater confidence that a vaccine will work, they begin to prepare for commercial launch. The entire process generally takes around 5 to 10 years. So, in the race of vaccine manufacturing, the old vaccine manufacturers were having advantage over the newer vaccine manufacturers because they are well aware of clinical trials, dosing, and other related things. The newer vaccine manufacturers will depend either on their own or onto their partners for help and understanding about the hurdles. So when there is a real pandemic will be arising like COVID-19 across the globe, so while manufacturing the COVID-19 vaccine, there will be a very limited preclinical testing, and there might be some sort of like, combining very small scale clinical trials for phase 1 and phase 2. Or they will directly omit phase 2 and directly shift to phase 3, where there is a interim data available, and simply they will establish the toxicological reports, and the lethal doses of the vaccine will be validated, so they can really launch the vaccine very soon. So it will be like around maybe a timeline of 6 to 8 months. Definitely in the case of such emergency, this sort of processes and this sort of like, bypassing the clinical trials like phase 1 and phase 2, and data related to is very much needed to save the lives, at least to have a better control over the pandemic. So while talking about different vaccine manufacturers about to manufacture like Sputnik, or in India there are like Covishield, Covaxin, which is a viral vector type of a vaccine, and many countries were approved to produce same vaccine to curb on the spread of corona pandemic situation. Across globe, there are many more vaccines are practiced to control the corona infection. However, we need to understand what sort of different vaccine coronavirus rather existed. So let's see how do different coronavirus. So basically, I would like to say that there is like a there is no laser pointer here. So I would say there are like a direct action and the kind of indirect action coronavirus is existed. In the case of the whole virus, which contains a weakened or inactivated version of corona or COVID-19 virus, and this will be purified with the help of chromatography and the chromatography resins. For those things will be definitely directly getting attenuated, so that they will have a controlled pathogenicity with the help of pharmaceutical excipients like preservatives. So, for example, the dosage formulations in the form of vials or in the form of pre-filled syringes will be manufactured, and those things will be directly injected into the human body. And because of that, once they will delivered into the human body, entered into the bloodstream, so that they will our immunity will take a control, and they will generate the anti-COVID antibodies. There is another type of vaccine where we need to take out the pathogenic pieces of COVID-19 virus or fragments of viruses, and similar dosage forms will be manufactured, and it will be injected into our bloodstream, and those fragments of viruses will start a immunogenic response into our body and will produce anti-COVID antibodies. So viral vector vaccines use a modified version of a different virus that we call as the vector to deliver important instructions to our cell. Firstly, the vector, not the virus that causes COVID-19, but a different harmless virus, will enter a cell in our body and then use the cell's machinery to produce a harmless piece of virus that causes COVID-19. This piece is known as a spike protein, and it is only found on the surface of the virus that causes COVID-19. In the next phase, the cell displays the spike protein onto the surface, and immune system recognizes it that it doesn't belong from our body. So this triggers our immune system to begin producing antibodies and activating other immune cells to fight off what it thinks is like an infection. At the end of the process, our bodies have learned how to protect against future infection with the virus that causes COVID-19. The benefit is that we get this protection from a vaccine without ever having to risk the serious consequences of getting sick with that particular COVID, any of the variants or any of the other different diseases. So any temporary discomfort experienced after getting the vaccine is a natural part of the process and an indication the vaccine is working. So we should not fear when we will have a fever, sorry. Talking about the mRNA vaccines. So mRNA vaccines are new, but not unknown. COVID-19 mRNA vaccines give instructions to our cells to make a harmless piece of what is called as a spike protein. As I've explained earlier, the spike protein is found on the surface of the viruses that causes COVID-19. Messenger RNA or mRNA is a genetic material that tells your body, okay, how to make proteins. The vaccine is made of mRNA wrapped in a coating that makes delivery easy and keeps the body from damaging it. The mRNA in the vaccine teaches your cells how to make copies of the spike protein. If you are exposed to the real virus later, your body will recognize it and it will fight off. I will be mentioning the process of unit operations which are needed to use it for the mRNA vaccine manufacturing in the coming slides. So because of these two types of vaccines, once they entered into the bloodstream, definitely they will be producing a protein that in case we call that as a S protein or spike protein, which will mimic like a virus pathogen and against which our immunity will produce anti-COVID antibodies. mRNA vaccines do not use the live virus that causes COVID-19. mRNA coronavirus is a kind of RNA or a nucleic acid vaccine based on mRNA technology. It mainly introduces the mRNA encoding virus S protein into human cells. Let human cells use the virus genetic information to transcribe and produce a novel coronavirus S protein so as to stimulate the body to start the immune response, not affect or interact with our DNA in any way. mRNA never enters the nucleus of the cell, which is where our DNA is kept. The cell breaks down and get rid of the mRNA soon after it is finished using instructions. Have a quick look onto the list of COVID vaccine developers and manufacturers across the globe. Almost all biosimilar manufacturers are leaped into the COVID-19 vaccine production. The vaccine categories include all types of vaccines which we discussed in previous slides like mRNA vaccine, non-replicating whole variant, viral vector, and there are some sort of like a nasal sprays are also available, will be available in coming days. Let us understand the common manufacturing processes involved into the vaccine, any type of vaccine manufacturing. So it always start with the upstreaming of viral cultures. And nowadays, either a stainless steel or single-use systems are involved into the upstream processes. The main aim of upstreaming or the upstream processes is amplifying the viral count without losing its native form of any of the microorganism or the viruses. Once the fermentation time in bioreactor is completed, we will bring that particular protein soup or viral soup into the further processing, and there might be some impurities. So we need to get rid of impurities, and because of that, the purification unit operations will pitch in into further purification processes. So the first step will be cell lysis, either by homogenization or by any of the other chemical processes, and then further we can get rid of cell debris with the help of continuous centrifuge or microfiltration with tangential flow filtration method. Initially, the volumes are very high, like maybe 5,000 liter or 10 KL, that is 10,000 liter. So to handle these huge volumes is definitely a big task. And to manage these volumes, we can further reduce it and simultaneously with the help of ultrafiltration or making a use of affinity chromatography, that is protein A or protein G chromatography. The another advantage of ultrafiltration is the another application called diafiltration. You can, in this diafiltration, you can do buffer exchange and create complementary work environment for further purification steps, and those are chromatography steps. After the couple of chromatography steps, the elution pool or the purified volume will be again concentrated as per the tighter concentration requirement, and also buffer will be exchanged as per the formulation dosage form, that might be a vial filling or pre-filled syringes filling. Then thus the purified drug substance which is formed, and that will be transferred to the fill and finish and further to packaging department. After the cultivation and the reproduction of coronavirus, it will be inactivated and then injected into the human body to stimulate our immunization. For example, in China, there are many biosimilar manufacturers as well who have leaped into the COVID-19 vaccine manufacturing and supplying them to different countries to have a better control and over the pandemic situations. To name the few are China National Biotech Group, situated at Beijing and at Wuhan, and there is a company called Sinovac are involved into the manufacturing of COVID-19 vaccine manufacturing. In the case of recombinant subunit protein vaccines, the COVID-19 vaccine that is made by the Novavax are developing is a recombinant subunit vaccine, meaning that SARS-CoV-2 components are created in the laboratory, okay, rather than isolated directly from the virus. Institute of Microbiology, Chinese Academy of Sciences at Beijing and Anhui, they are also involved to produce the recombinant protein subunits, which will mimic like a coronavirus and the fragment or some pathogenic part of it, which will give a immunogenic response into our body. So talking about again the recombinant protein vaccines are so they are like a car which would carry an antigen gene of COVID-19 or any of the the viruses, and after reaching to the human cells, antigen gene of that particular virus. In our case, we are talking about the COVID-19 virus will not be reproduced and has no pathogenicity, but it could express the protein antigen of COVID-19 to stimulate human immune system to generate the antibody. For example, the AstraZeneca comes with this type of vaccine and approved many countries to manufacture like UK, China, India, and United States of America as well. In China, Convindicia Casino Bio also manufacturing the recombinant adenovirus vector form of vaccine. Okay, so now, after giving the brief introduction about the corona, different type of vaccines, let's see overview of the production of processes, whatever we discuss about the COVID-19 vaccine manufacturing. So this slide shows the manufacturing of inactivated slides. The drug substance is created by firstly finalizing the cell lines, and there will be a virus inoculation and fermentation of those viral culture will happen in bioreactors, as I mentioned earlier, and further those are purified with the unit operations like ultrafiltration and chromatography. Further, the purified drug substance will be subjected to filling lines like the ultrasonic cleaning, air injection, and to the pre-filled syringe filling lines. Those things will be again transferred to the packaging lines, and TRUKIng technology will help you to provide the filling lines and packaging lines as well. Likewise, we have just seen that about there are like inactivated vaccine manufacturing, the upstreaming of vaccine manufacturing will be different, and rest of the process will same. That will be like a downstream processing, and then the drug substance will be transferred to the filling lines and further for packaging and quality testing. So in the case of mRNA vaccine production, as I have mentioned, we need to select the target messenger RNA, which will eventually give immune response of S protein production inside the body. Then this particular mRNA will be amplified into multiple copies of it, and further those will be purified with the help of ultrafiltration and chromatography steps, and after that it will be transferred for liquid inorganic nanoparticle encapsulation technology to form the nanoparticles. The mRNA technology required low temperatures to operate, and then rest of the process of filling, packaging, and the quality testing will be carried out. So TRUKING technology is providing one roof solution for entire process. Let's have a quick look onto the entire process and offerings from TRUKING technology. So talking about the overall process for vaccine manufacturing, there are different stages like media preparation and choosing the cell lines, creating the seed, and further to the seed amplification, or the in case of like any of the like E. coli amplification, or the viral cell culture amplification. Various size bioreactors we provide, and that will be included into like stainless steel bioreactors, or we are very well equipped and catering to the biopharmaceutical company with the single-use bioreactors as well. Also, we need to make some buffers for the preparation of different compositions for various applications. We do have the mixers, and the depth filtration systems are also provided for the further application of liquid handling. Once we have done with the seed bioreactor, then the cell this will be amplified, will be transferred to the bigger bioreactors, that is say about like 2000 liter to 5000 liter bioreactors. After the fermentation process is completed, as I said, it will be subjected to for the further clarification and for the purification steps. Overall, TRUKING technology will provide you in all aspects of the process. It will right away, it will start from choosing the cell lines, then the media preparation, or for the purification of the impurities, filling lines and packaging lines. Also, whether it will be an ampule line or a pre-filled syringe. I wanted to mention that the bioreactors or the chromatography system is provided by the TRUKING technology, can be supplied in terms of like the stainless steel, or in a the collapsible single-use bags. Now let's study a case study for like TRUKING technology. Let's take an example for the vaccine manufacturing, where there will be an upstream cell culture is required, downstream purification, buffer preparation, and production preparation and filling. TRUKING technology not only provide the hardware, but also along with the process intensification equipments and the technical support, TRUKING technology will also support you for your greenfield projects. In this project, we will guide you not only for the technical processes, but also the entire erection of biopharmaceutical manufacturing facility. TRUKING will support you for the layout of your floor designs and optimum use of the facility. So this line talks about the timeline required for each unit operations which are involved into the for upstreaming to the drug substance transfer. So initial inoculation, it will take around 2 to 7 days to grow into a good number. For further cell amplification into a seeding stage, it requires around 4 to 7 days, and the cells which are amplified that will be put for the production scale, it will be around 7 to 9 days of time, or it depends on the cell line whether it will go for around 14 days as well. Once this upstream batch will be completed, then at least around 2 to 6 days of time required as per the batch size and the level of purification. And then the drug substance so produced will be transferred to the filling lines and packaging lines. Now, while doing the upstream or the downstream processing, we need media. So media and buffer preparation are the basic unit operations in any biosimilar manufacturing, not in the case of only in vaccine manufacturing, but in the case of maybe a monoclonal antibody or insulin production. So we need media and the buffer preparation there. In case of cell seeding and the amplification, which also requires a kind of media components and sodium hydroxide or sodium bicarbonate like solutions. For virus amplification, they require glucose solutions or acidic and basic solutions and also like the surfactant like twin 80 solutions. For further processing of cell cultures like cell lysis, enzyme treatment, clarification, and CIP, that is the cleaning in place for bioreactors as well as for the downstream equipments, we need buffers and CIP solutions. While talking only about this cleaning in place, that is the CIP, we widely use sodium hydroxide, which is cheap and very effective. So continuing with the buffer preparation, likewise in upstream operations, we need buffers to be prepared for downstream operations as well. For example, there are various phase chromatography, and every phase of chromatography needs specific buffer requirement. For example, each chromatography run will have a different phases like equilibration, sample loading, post-load wash. This post-load wash may involve wash 1 and wash 2, and further with the elution where your impurities and your protein of interest will elute out, and then followed with the cleaning in place of that particular chromatography resin. So in the case of media preparation, what we need to do is we need to weigh the media components, or their nowadays the premixes are also available and provided by the vendors for your processes. Then we will dissolve into those things into a distilled water or sterilized water, and it has to be all the components should be in a sterilized conditions. Once we have the sterilized media, we can put the cell culture media for the storage, or we can pass it on to the reactor for the fermentation processes. So TRUKING technology will guide you, and we do have a media preparation process equipments for your better use, so that there will not be much of the time. It will not be a very time consuming processes, and you can have a worry free for your media preparation for every batch. Same like media preparation, we need to prepare the buffers either for upstream or the downstream processes. The same process will be there when you will weigh your the buffer components, and then you will again dissolve into the deionized or the sterilized water. And to avoid further issues of increase in pressure, it is better to get the buffers filtered through the microfiltration units, and as and when needed, either you can store it, transfer to the above tanks, and carry out the operations. So the tough point in the buffer preparation processes are mainly like maintaining the media buffer preparation tanks and cleaning them and sanitizing them as well. It is a time consuming process. So these are the major hurdles and day-to-day activity, and it will definitely consume a lot of time and resulting into the less onto the productivity of your facility. From TRUKING technology, what we can offer you the process intensification equipment, that is the buffer preparation hardware like liquid dosing system and inline conditioning buffer preparation systems. There are hybrid technology like inline conditioner buffer system is equipped with the column walls and carry out the function of chromatography system on these instruments. I will be talking on these equipment in coming slides. In the case of upstream operations, we always start with the seeding of your cell culture, and the seeding purpose, either you can use your flask, a smaller flask, or you can use a different bioreactors. For example, a small bioreactor of like 5 liters, or you can use a rocking platform where the 50-liter bioreactor you can amplify your cell culture up to 200 liter, and then further you can inoculate your cell amplified for the production batches. So these bioreactors, what TRUKING is providing is range from 20 liter, 50 liter, 100 liter, and 200 liter are available for seeding purpose. As per your requirement, definitely you can make a use of it. In the case of further operations, when we look at it, those are mostly non-specific or specific purification technologies. For example, ultrafiltration and chromatographic operations. So for the ultrafiltration, TRUKING technology is offering the tangential flow filtration hardware systems where as per your need, you can attach a micromembranes or the ultra micromembranes for the microfiltration purpose and ultrafiltration membranes for your ultrafiltration purpose. Also, along with that, these ultrafiltration and the depth filtration hardware is very much compatible with the various vendors of membrane cassettes for the cross flow filtration fiber. These things you can easily fit it and you can carry out the auto operations as well. In the case of chromatography, systems, you can combine the chromatography system with the chromatography columns, and you can carry out a purification operation. In the case of chromatography system, we do have a different, the flow rate variations. For example, it will start from 180 liters to 2000 or maybe like 2400 liters per hour. Recently, in a special case, we have provided around, like a 12,000 liter per hour system in China as well. In the case of autocolumn, the diameter range will start from 30 centimeter to 1.6 meter. That is, the 1600 millimeters of a diameter. You can use with our autocolumn. And to pack our autocolumn, you need a packing station where one packing station can be utilized with a couple of more columns of different diameters. So as well, along with the packing station, TRUKING technology can also provide the slurry tanks with the alpha helical structure impeller inside, so that there will be a minimum stress onto the chromatography resin beads, and there will not be any attrition of the chromatography beads because of helical structure of the impellers, and you can keep your slurry rotating in a homogeneous suspension form. In the case of wave bioreactors, the inflated single-use bags will be mounted onto the wave bioreactors, and the cell culture media will be filled with the cell culture media and will be kept in rocking position up and down. And you can have a different sensors like pH, dissolved oxygen, temperature, and the other sensors. It can be controlled with the help of software. TRUKING technology can offer you wave bioreactors from 5 liters to 500 liters. And also in the case of bioreactors, you can manage your rocking speed and angle of rocking as well. Apart from these things, you can control your temperature and the ventilation, and you can have a better control onto the weight of the bags and the media and couple of other parameters you can definitely control. In the case of upstream processes, seeding and further production of scale, TRUKING technology can support you with stainless steel and single-use bioreactors. So depending on the production goals, the scientist or the end user can now cycle between a batch mode or a fed batch or a perfusion bioreactors modes. So these TRUKING bioreactors provide you a scalable and a robust stir tank performance up to around 5,000 liter in both CGMP and non-CGMP environments. These bioreactors are very reliable and quantifiable for the process performance. These are very, you can see these are in modular in design, enables configurability and provides added flexibility also. These are very much adaptable for various molecules and applications of or multi-drug processes. You can easily scale up those reactors and you can predict and seamless You can scale it up from like, maybe like 2000 to 5000 liters easily. So these are the exact specifications of bioreactors from 30 liter to 5000 liter. In these bioreactors, with the help of software, we can better control over the speed of aeration or gases, and pH and temperature sensors are also available for accurate measuring. We do have the defoaming electrodes, which will detect the foaming or frothing happening inside the fermentation processes. The conditioning and the preparation tanks can be utilized in various applications, like used in cell culture media preparation, buffer preparation in upstream and downstream processes, in semi-finished products as well. So these conditioning and preparation tanks are enabled with the the load cells, which can definitely give you a better idea about the weight of the liquid, and also they are enabled with the temperature control, and you can attach CIP and SIP systems for further sanitization purposes. So these media preparation tanks can range from 200 liter to 1000 liter, and storage system can range from 50 liter to 1000 liter. After the cell harvesting and the cell lysis, the fermented soup will be further clarified with the help of depth filtration. TRUKING Technology is offering you the hardware for depth filtration systems, which are enabled with inlet pressure monitoring to control the delta pressure, which eventually controls the pressure coming from the storage tanks or the preparation tanks. It will control the flow of liquid at inlet and avoid siphoning of effect of the fermented soup. Along with that, the depth filtration unit also has a constant flow filtration, and constant pressure flow options are also available. Depth filtration clarification systems can be maximum operated at 6 bar, or approximately in 87 psi pressure units. So depth filtration modules you can attach, for example, 10 to 20 meter square depth filter, and you can easily have a load capacity of 50 to 100 liter per meter square. The flux can vary between 50 to 100 liter per meter per hour, and yeah. Again, for the concentration or the dye filtration, that is the buffer exchange, and for some non-specific purifications, we can use ultrafiltration unit operations. TRUKING Technology offers a state-of-the-art equipped with equally good software, which have better controls over the constant flow, operations, or you can also monitor the constant pressure or the constant flux. So whenever there will be a fouling of the membrane modules, you will be alarmed or notified. Also, along with the constant flow, constant pressure and flux, you can also keep an eye onto the constant transmembrane pressure for your better control onto the permeate which is coming out of the ultrafiltration modules, maybe like a hollow fiber or a cassettes. So this ultrafiltration unit can maximally operated for 6 bars, which is, I think, is a very good the pressure limit for membrane assemblies which are available in this particular market. For this unit, ultrafiltration unit, you can either fit the membrane cassettes or the hollow fibers. The chromatography systems from TRUKING Technology is another flagship product offering for biopharmaceutical products. So we have been installed these chromatography systems like around many systems in India as well. For example, the Serum Institute of India, then the Bharat Biotech, Dr. Reddy's, and many more biopharmaceutical companies here in India. These chromatography systems will start with various flow rates as per the customer need, from around like 180 liters to around 2400 liters per hour, as well as we can build a specific chromatography system as per your need as well. The maximum operating pressure for this chromatography system is again 6 bar. This chromatography system comes under two variations like single-use flow paths and stainless steel flow path. As per your need, we can provide you those things for your application. So when this chromatography system will be a single-use flow path, then definitely a same system can be used for the multi-product facility in a very less turnaround time. TRUKING chromatography systems are equipped with operated through the PuriCat software. This PuriCat software is 21 CFR Part 11 compliant, and which means that there will not be any tampering happening in any of the run logs which are captured, and this is very much important auditable issue for any of the manufacturing projects. If you see this PuriCat software screenshot, you can see that it is very intuitive from right from the there are like the inlets like SA1, SA2, SB1, SB2, and there will be two pumps, that is pump A and pump B, and the flow path you can see that the bubble trap, and there is a couple of sensors like pH sensor, pre-column pH sensor, and the conductivity sensor, pre-column pressure sensor, and these sensors are definitely attached. And then there will be a column where you will have the options of like a downflow and upflow, and after the column there will be a post pH, post pressure, and post conductivity, followed with the UV sensor is available for your illusion purposes. As you can see, this is interactive area where there are different regions, the legends like UV1 and UV2, and there might be a conductivity or pH. The actual chromatogram you can see in this thing, which is exactly happening at a particular time interval. The previous runs, which can be overlapped with the current chromatogram, so that we can usually we can see how our run is progressing and what will be the conductivity of the pH, or any of the changes occurring in the runs which happened previously and into the current runs. We can easily compare, so that we can take a better control of your the chromatography run. In this PuriCat software, we do have the basic settings where you can ask for your, as per your choice, you can turn on or turn off the walls or any of the sensors if you wanted to have a specific need for your particular run. The column diameters and the bed height, if you put over here, then you can easily calculate your column volume. And then we do have the alarm settings, where we can enable with the specific values like the pre-pressure high alarms, pre-pressure low alarms, and you can put different range of values and you can for a particular region like a temperature alarms or the air pressure or the air sensors. So you can put your values, and so that after giving these alarm situations or the commands for these alarms, your chromatography run will be under a continuous watch unless and until it will complete. And by surpassing the this alarm commands, it will definitely intimate you that there will be a high pressure or a low pressure, and you can take a better control of your systems. So these pre-settings where you can actually keep walls open, and where you can if you want, you can further keep and check on to whether to open which wall when you are going to start your system. So there are multiple the options are available in the PuriCat software. And talking about the best feature is the method construction. So you can create a single method on the basis of like there are different phases you can create, like equilibration, then the sample loading, preload wash, elution, cleaning in phase. So we can construct these methods, and you can save it, so that whenever you wanted to go for a next run, you just pull a particular method, and then you can quickly start with the better control of your processes, and all alarms and everything will be mentioned in this particular method. and then you don't have to worry about like every time any of the alarming situations because of the method construction. Talking about the post run, that is the peak integration is another parameter which is widely operated by the end users. So we do have all sort of operations and can be operated to the peak integration module, where you can have you can choose your curve, and there will be a different peak integration options are available, like splicing, or you can zoom in, you can splice it up, you can compare, you can further quantify the data by on the basis of the standard curves, and all whatnot. All options will be very much available in our PureCAD software. Once we have a good chromatography system, then definitely for the purification purpose, we need to have the chromatography column. So the auto column chromatography columns from TRUKIng Technology is another flagship product which is attached to the chromatography systems to carry out one complete chromatography run. These columns comes with various dimensions. For example, these columns will start from 30 centimeter, that is 300 millimeter, to 1600 millimeter, that is 1.6 meter in diameter, and there are different column tubes are available like 550, 750, and 1000 millimeters in length. These columns will be operated from the range of 5 bar to 4 bar independently, like onto the diameter of the column. So these columns are having a patented mechanical structure design, where the triangular mechanical structure tendons the overall strength of the equipment and makes the equipment simple, lightweight, and safer for use. The design pressure for this particular column is around 5 bar. You can see over here the traditional design of the same range of column from different vendors, where the top adapter is operated through the central shaft, which will not only having the imbalance across the higher diameter of the columns. For example, because of there is an imbalance, there is no millimeter accuracy is you can achieve or you can observe. While in the case of auto columns, there are 4 points holding the top adapter, and which facilitates the up and down movement of the top adapter, giving the perfect horizontal position, and which results into the millimeter accuracy for these auto columns. So these auto columns have synchronized servo motors, two servo motors, which controls up and down movement of the top adapter. These auto columns also adopt the hoisting method on its own. That means you don't need any extra space for the maintenance activities. Because of the hoisting method of this auto column, which occupies a very small area and resulting into your saving and manufacturing space, and it is very much convenient for the transportation, and it only takes around 15 to 20 minutes to disassemble the entire auto column. While in the case of traditional design, the column tube will swivel out, and because of that, you need to keep an extra space for the maintenance activities, and it requires a little more time for disassembling the top and bottom adapters. Just to check the flow distribution inside the column, we have given the multiple red dye 5 to 6 injections into the equal time interval, and the flow was given from upward towards the downward, and you can see that for after every injection there is a very solid and compact red ring is formed. Just to check the inside flow distribution, we have scooped out the cake, and we found that there is a very compact formation of red dye color, and which for every pulse of a red dye injection given at a particular time interval. And also you can check the column qualification criteria, where there will be around 7,400 plates per meter, and the asymmetry value is 1.07, which is of very high standard value for the chromatography column qualification. We have used JNC, a company from Japan, the chromatographic media from them. The name of the media was Cellophane GCL 2000. The auto column from TRUKING Technology is a combination of two columns, that is pack in place and also a syringe action axial compression packing of resins. As per the client requirement or the chromatographic resin demand, we can use a pack in place method, or you can also pull the resin with the help of the like a syringe action and a resin slurry while doing the upward direction movement of top adapter, and then again compress the resin by bringing back the top adapter and towards the down position with the further compressing of the resin bed and achieving the desired bed height with the millimeter accuracy and accurate compression factors. Along with these chromatography systems and chromatography columns, we also have the slurry tanks for homogeneous mixing of slurry and for the packing of chromatography columns. We are using an impeller in the slurry tank is a helical structure, so that there will be a minimum stress given to the beads and the cell lines as well, and still the resin will be in a suspension form in a homogeneous situation. As per need, we do have the different volumes. For example, we can supply you around 1000 liters volume for the slurry tank. These tanks come up with the CIP and SIP-able option and have a proper documentation for the audit purpose. So overall, TRUKING Technology is providing you a unique combination of state-of-the-art process intensification equipment for bioprocessing projects, which includes upstream and downstream operations. Along with the bioprocessing unit operations or the equipments, we have imminent presence in the liquid handling dosage form systems like vial or a syringe dosing or auto inspection of vial and syringe machines are also available, and packaging lines along with the lyophilizers are also available. So these are our biopharmaceutical clients across the globe to whom we are partnering and catering for the different biosimilar or vaccine manufacturing in their manufacturing plants. So thank you very much, guys. If you have any questions, I will be very glad to answer your questions.
Thank you very much. Very, very interesting. The first part in which you explain all the technology, well, all the virus types, and then all the process, and of course all the machines and that are included on that you need to follow on the process. We are short of time, but we still have 4 or 5 minutes. Tenemos 4 o 5 minutos por si hubiera alguna pregunta que gustarían que tratáramos de resolver, ya sea en TRUKING o Mr. Pramod. Y me gustaría nada más rápidamente, se me pasó y se nos pasó ahí en TRUKING. Mr. Pramod ganó el premio de Best Innovative Solution en el 2018. Es importante mencionar esto, como lo vieron en su presentación bastante robusta. Entonces, pues bueno, muy profunda todo el análisis del proceso que hicimos. Nada, me quería hacer eso. No sé si hay alguna pregunta o alguien que quiera comentar algo. Tenemos mucha gente. No sé si por ahí alguien tenemos en el chat algo. O Sabrina, si por ahí en TRUKING nos quieren comentar algo.
Yeah, guys, any questions?
I'm looking if there's any question, Pramod. Let me see if on the chat or someone else. Okay, no. Okay, here acabamos de lanzar una pregunta, si nos pueden ayudar rápidamente para ver los resultados y poder mandarles más información.
Y también les agradezco mucho si pueden hacer las operaciones que presenté en la presentación. Por favor, muchas gracias. Y también gracias por participar a esta presentación y gracias por su tiempo. TRUKING ofrece soluciones completas de equipamiento de proceso y empaque para los laboratorios farmacéuticos mundiales. Si tiene algunas dudas o tiene intereses conocernos más, por favor no duden en contactarnos y Grupo Rush. Muchas gracias y que tengan un buen día.
Okay, only one question, Pramod. Cesar is asking the main goal of the sensors, which is the main goal or the main of the sensor for pH and ORP. I don't know what means ORP. The main challenge, sorry, on this technology. I don't know if
I didn't able to get you, sir. What is the exact question?
it says using the SST technology, which is the main challenge of the sensors of pH and ORP on this technology. I don't know.
The most challenging is the gaseous exchange when we the sensor is like measuring how the gas is diffusing and those things. So pH and everything is very normal. We will have the exact pH measuring. See, for hardware part is not a issue, but definitely as the process goes on, and whenever the bacteria or any of the microorganisms secrete any of the protein or the protein is expressed, that time definitely there will be a slight pH change. But we are definitely taking care of the pH and other the conductivity with the help of our the solutions like any of the other buffers, which we try to continuously feed in to the bioreactor so that we can keep a pH over there. So that will not be a major issue.
Ok, César, no sé si te quedó. Ok, ya vi que te quedó claro ahí la respuesta. Por ahí la presentación, vamos a platicar con Truking si podemos compartir algo de esta presentación. Si no, de todos modos, vía Truking, vía nosotros, vamos a poder compartir algo de información sobre esta tecnología y los procesos. Ya no sé, el tiempo, ya estamos sobre el tiempo. No me gustaría robar más del tiempo que ya nos cedieron. Creo que si no hay más, no me toca más que agradecer, Mr. Pramod, for this nice presentation and all your knowledge that you share with us. Y a todo el grupo, muchísimas gracias. Estamos terminando el primer día de FEVIE. Muy exitosos, muy contentos, muy animados de que estén todos aquí con nosotros. Tenemos todavía 3 días por delante. Con otras pláticas en otras industrias de automotriz, vamos a tener algo de biodescontaminación y vamos a seguir hablando de las vacunas. Entonces, ya para finalizar, acabamos de lanzar la evaluación del webinar. Si nos pudieran ayudar, todo esto nos sirve para mejorar y tratar de, por favor, mejorar y hacer las cosas mejor. También Esther compartió en el chat el resto de la agenda del FEVIE. Estamos hasta el viernes a mediodía. Sí, la verdad es que todavía hay temas muy, muy interesantes. Tenemos algo de máquinas. Mañana Romaco se presenta. OMA, también vamos a tener algo de biodescontaminación. Vamos a seguir hablando de arquitectura de cuartos limpios con una tecnología de las puertas. Vamos a hablar de un webinar que por ahí muy, muy interesante, que nos preparamos para todos ustedes, que es sobre servicio postventa, mantenimientos preventivos, con el equipo de David, que aquí nos acompaña, Yolanda, Martín. Les tenemos ahí una sorpresita para terminar el webinar del FEVIE el viernes. Si ya no hay más, no me queda más que agradecerles. Por ahí veo gente de Nicaragua, gente de Centroamérica. Muchas gracias por conectarse también. Y ya saben que están abiertas nuestras tecnologías vía nuestros, vía aquí las personas de ventas. Y si no, con todo gusto los canalizamos. No sé si hay algo más. Sabrina, gracias por todo. Mr. Pramod, thank you very much. And,
thank you, sir. Thank you very much, everyone. Very much. Gracias.
Gracias.
I'm sorry, I'm very less with the Mexican language or the Spanish language.
No problem. Your English was very, very clear. Y por ahí vieron que había los subtítulos en inglés simultáneos, eso ayuda bastante. Y gracias, amiga, gracias, thank you to the trucking team over there. Y nos estamos viendo. Buena tarde y saludos. Gracias.
Gracias por todo.
Buena tarde a todos.
Thank you very much. Thank you. See you
Gracias,
soon.
César.
Gracias, buena tarde.
Ahí nos vemos.